Hello, my name is Yuting, and here I will talk about why trees trump trees for ancestral genome reconstruction. In ancestral reconstruction, we are given a gene family or all gene families in present-day species, and the goal is to determine what genes were present in ancestral species. This also tells us how many gene copies were in the ancestral states. Therefore, reconstruction of ancestral gene content can reveal the age of a gene family, the evolutionary processes that shape the genome, the expansions and contractions in the functional repertoire encoded in the genome, and the relationship between changes in genomic coding potential to biochemical, developmental, and morphological innovations. Here, we compare two parsimony-based ancestral genome reconstruction methods: Wagner parsimony and tree reconciliation. Wagner parsimony was originally designed to capture the gain and loss of phenotypic traits, such as morphological features, and was later adapted to capture the gain and loss of genomic traits, like genes, folds, and introns. Tree reconciliation reconstructs ancestral genomic states and events by fitting a gene tree into a corresponding species tree and minimizing the weighted gene duplication, transfer, and loss events. Wagner parsimony takes sites of gene families in present-day genomes as its input and reconstructs ancestral states that minimize gains and losses. However, Wagner parsimony uses implicit model and cannot distinguish between transfers and duplications, which means it is challenged by parallel gains, parallel losses, and reversals. For example, if the true history contains one horizontal gene transfer and one independent gain event, Wagner parsimony would not be able to recover that. Reconciliation indirectly takes sequencing information into consideration by using gene trees as the input. The reconstruction, constrained by gene tree topology, reconstruct ancestral states that minimize duplications, transfers, and losses. While both methods use a parsimony approach, reconciliation uses an explicit event model that can distinguish between the types of gain events that contribute to the expansion, while Wagner parsimony cannot. Moreover, because Wagner parsimony only uses gene counts, it also minimizing parallel gains, parallel losses, and reversals, and consider them as rare events. Given that horizontal gene transfer can mimic parallel gains, Wagner also considers those rare. Signal from the gene tree topology may reveal those types of events with reconciliation. We predict that reconciliation will outperform Wagner parsimony when horizontal gene transfer is a frequent event. Here provided a mini example of the reconstructions of a species tree with a gene tree. Reconciliation infers one duplication, one horizontal gene transfer from C to T, and one gene copy in the ancestral species P. Wagner parsimony infers one early gain instead of two parallel recent gains, and two copies in the ancestral species P. To test our prediction and better investigate the differences between reconciliation and Wagner parsimony for ancestral reconstruction in real data where horizontal gene transfer is frequent, we reconciled over 10,000 gene trees sampled from cyanobacteria species using Notown. The results wasn't compared with ancestral gene counts inferred with Wagner parsimony using count. And for now, we will focus on this part of the tree. This figure shows the ancestral gene counts inferred using two methods. Each present-day species is labeled with the number of encoded genes that are members of the 10,000 families used in this analysis. The internal nodes are labeled with the inferred gene counts in ancestral genomes. On each branch, the number of gains are annotated with the blue bar, and losses are annotated with the ivory bar. Looking at the left-hand side first, we can see that Wagner parsimony infers a gradual expansion, a genome size from 2,500 in node N45 to 4,000 genes on average in present-day genomes. Also, on each branch, Wagner parsimony infers many more gains than losses. In contrast, reconciliation infers rapid expansion to the present-day genome size, followed by specialization and genome streamlining. Also, the numbers of inferred gains and losses are balanced in many branches, which suggests constant turnover of gene content during evolution. Here is a side-by-side comparison for the inferred gains and losses of genes on each branch. We can see clearly that the numbers of gene gains and losses inferred with Wagner parsimony and Yuting differ dramatically on each branch. Figure to the left shows the total gains and losses comparison between Wagner parsimony and Yuting. Again, gene gain dominates gene loss in Wagner. To sum up, our results demonstrate how reconciliation and Wagner parsimony can lead to very different ancestral reconstructions and very different conclusions about the processes driving genome evolution in this species. In this dataset, Wagner parsimony is clearly challenged by the horizontal gene transfer. This illustrates the dramatic impact of the strict assumptions of Wagner parsimony criterion. In comparison, Yuting benefits from greater information available from using gene trees and an explicit event model that distinguishes between duplication and horizontal gene transfer. Therefore, Yuting can inform more nuanced evolutionary history. Thank you for your time, and thank you for your interest.